Welcome to The Explainer. Today, we're embarking on what I honestly consider to be one of the absolute most incredible journeys in all of biology. We are going to look at the literal physical journey of a memory. It's so easy to just think of human memory like a digital hard drive, right? Just data floating around in a cloud somewhere inside our heads. But the reality, it's far more fascinating than that. Every single thing you remember is a living, breathing, physical change inside your brain. It's literally structural engineering on a microscopic scale, and today we're going to look exactly at how your brain builds those structures. So think about one of your strongest memories for a second. Maybe it's a mental photo from a family gathering years ago that just brings a rush of emotion flooding back. Or think about the process of learning a complex new skill, like driving a car or picking up a musical instrument. How does a totally fleeting experience, just a quick sound, a sight, a feeling, literally rewrite the physical hardware of your mind? Well, to really understand this, we have to start at the foundational building blocks and work our way up. Okay, let's dive into this. Here is our visual roadmap for today's explainer. Number 1, the architecture of memory. 2, microfactories in your synapses. 3, choosing the memory engram. 4, connecting location and reward. And finally, 5, the overnight memory shift. Kicking things off with Section 1, the architecture of memory, the brain's vast network. 100 billion. Just wrap your head around that number for a second. That is the estimated number of neurons, the fundamental processing cells in your brain. It is this incredibly massive, interconnected network of arguably the most remarkable cells in the living world. Each of these neurons has a long fiber called an axon that branches out into these tiny terminals, basically reaching out to touch its neighbors. And it's exactly at those microscopic meeting points where the magic of memory actually happens. And that meeting point, that's called a synapse. Technically, a synapse is the tiny gap or junction where specialized proteins allow electrical and chemical signals to pass from the axon of one neuron right over to the root-like dendrites of another. Over time, as you learn and experience the world around you, these connections are either physically strengthened or they weaken and eventually fade away entirely. So memories are literally built by strengthening these tiny synaptic connections. But as you'll see, making those connections stronger presents a massive biological challenge. Which brings us to Section 2, microfactories in your synapses, local 3D printing. So here is what we call the memory problem. To strengthen a synapse, you absolutely need new proteins. Now, in most normal cells, proteins are manufactured centrally, right in the main cell body. But neurons are incredibly long and extremely thin. A single neuron might have thousands of synapses, and each one of them needs its own specific proteins on demand to actually form a memory. If the neuron had to assemble proteins in the central cell body and ship them all the way down the axon, it could take hours. And by then, that fleeting moment you were trying to remember is already gone. The brain absolutely needs instant memory creation. So nature came up with just a brilliant solution. It essentially set up local 3D printers at every single synapse. Here's exactly how it works. An experience happens, but instead of sending finished proteins all the way down that long axon, the brain sends messenger RNA, mRNA, which acts as a localized set of instructions. This mRNA just waits right at the synapse. Then, right on demand, the proteins are synthesized locally, exactly when and where they're needed. And get this, researchers recently made a massive breakthrough here. They found that an enzyme releases a saturated fat called myristic acid. It acts basically like a cellular superglue, hooking onto those newly synthesized proteins to firmly lock the memory in place. If scientists can eventually master this microscopic glue, we could potentially treat things like PTSD by preventing traumatic memories from sticking, or even combat Alzheimer's by helping fading memories hold on. It's absolutely game-changing. Let's move to Section 3 and see how this all builds, choosing the memory engram, selecting the active cells. Now, to figure out how the brain chooses which cells to actually keep, researchers used a cutting-edge optogenetic tool. Think of it like a highly precise cellular spy gadget called FF-Liker. It uses high intracellular calcium and light to basically tag cells with unprecedented, down-to-the-second precision. So they studied mice during a learning exercise called fear conditioning, which is essentially teaching the mice to associate a specific environment with a very mild shock. Because this tool is so unbelievably fast, researchers were able to categorize the mice's microscopic brain cell activity into 4 highly specific behavioral groups: cells active before the shock, cells active right during the shock, cells active while the mice were freezing in fear, and cells active when they were not freezing. So the crucial point the study revealed is that if you later artificially reactivate those pre-shock cells, literally nothing happens. The brain actually discards those initial contextual cells altogether. It is only the cells that were active during highly salient emotional states, those exact moments of shock and freezing, that are selected to become the permanent memory engram. Basically, the brain filters out all the background noise and physically builds the memory exclusively out of the neurons firing during the most impactful parts of the experience. Let's zoom out a little further for Section 4, Connecting Location and Reward, the nucleus accumbens. A really fascinating recent study from the University of Maryland tackled this by elegantly contrasting two totally distinct inputs in the brain. On one hand, you have the dorsal hippocampus, which acts pretty much like your internal GPS, handling your spatial memory, the where of a situation. Then you have the ventral hippocampus, which is heavily tied to emotion and motivation, the why it feels good part. Now, for a long time, scientists assumed these circuits were mostly separate. But think about it. To navigate to your favorite restaurant, your brain has to seamlessly combine the map with the craving, right? So, using ultra-high-resolution digital slices, researchers discovered something absolutely incredible. These separate signals actually converge on the exact same individual neurons in a specific region called the nucleus accumbens. The synapses from the where pathway and the why pathway literally sit mere microns apart on the neuron's dendrites. And because they are just so incredibly close to one another, when both inputs are active at the exact same time, they massively amplify each other. It's a truly beautiful illustration of how the brain seamlessly weaves together memory and motivation at a microscopic level to drive our everyday decisions. Which brings us finally to Section 5, the overnight memory shift, a change of guard. A really fascinating study out of Singzhou Hospital monitored patients over 6 days and managed to chart this incredible biological shift of guard. Here's how it works. On day 1 of learning something totally new, the hippocampus, which is the brain's fast-acting memory center, is the commander. It leads the whole process through synchronized neural activity. But overnight, while you're fast asleep, this quiet transfer happens. By day 2 onwards, the primary control of that memory completely shifts to the neocortex, which is the outer layer of the brain responsible for long-term storage and much higher cognitive function. And this brilliantly illustrates the point. As lead researcher Wang Lukong notes, the process can be compared to learning how to drive. On the first day, a driving instructor closely guides every move. After a good night's sleep, however, the brain's cortical networks take over. This shift is exactly what allows the new skill to become fully automatic and structurally stable. So what you really should take away from this is that sleep is absolutely not just passive downtime. No way. It is an active, deeply essential biological mechanism. It is the vital final step required to consolidate all those microscopic synaptic changes we've been talking about today, physically moving them from temporary storage into the permanent structural architecture of your mind. Now, what's really interesting here is the profound realization this leaves us with. Every single experience you've had today has sparked local 3D printing in your synapses. But it's tonight, when you finally close your eyes, that the real structural engineering happens. The driving instructor hands over the keys, and your memories literally become a permanent physical part of who you are. Thank you so much for joining me on this incredible journey through the brain. If you enjoyed this explainer, please make sure to like, share, and subscribe, and hit that bell icon for more deep dives. And hey, if you are ever in need of physical therapy or emergency pain relief treatments, be sure to visit the Dr. Gangadhar Physiotherapy and Rehabilitation Center. All their contact info is available right in our presentation. As we sign off today, I want to leave you with this thought. Knowing what you know now about this microscopic structural architecture, what physical structures will your mind build tonight? Keep learning, and I will see you in the next explainer.